Okay, so I posted this announcement the other day. It should have come into your email as well. And so with the exam coming up on Thursday in your lab section, and it's going to start with the plant growth. So the plant growth slide set, when you get down toward the end, so essentially this, I think it's slide 50 or 51, at least that's what it looks like on my end. So this section forward in the plant growth slide set. And that's where the information on phytochromes is located, right? So that portion of plant growth slide set, and then we get into photosynthesis, and then so that whole slide set, and then the roots and stems in lecture. That's what we're covering. We will not cover leaves in lecture. But you did have a chance to do that a little bit in lab, so there'll be a couple questions in lab on leaves. So let me go back to any questions on the exam content? Also, the assignment that was posted in EDUCAT that I pushed to a Saturday deadline because of the snow day. So I've released the scores on that and the feedback. So please look at that. If you see something wonky, like, hey, I answered that correctly, but it scored me incorrectly, send me an email and let me know, because every once in a while that will happen where there's a little glitch in the system. The C3 cycle. So the Calvin cycle, looking at that. And so we're thinking about the stomata opening up, CO2 coming in, and then it reacts with RUBP. So the first stable product is the PGA molecule, phosphoglyceric acid, PGA. Yes, because if there's a minor spelling issue or something like that, and the computer is very strict, you know, and so if you tell me, I can go in and manually grade and make the change. So please look over your answers and what the correct answer is. Send me an email if you see it, just, you know, hey, I did answer that correctly. Maybe I just switched out two letters or something. So let me know. Okay. I caught some, but I haven't gone through every single person yet. So I'm making my way through it. Okay. So I wanted to talk, there's a few last things I want to talk to you about in the STEM slide set. We didn't really talk about product yet, so I wanted to talk about that. And then we'll get into leaves today as well. So here's a question for you. So we've looked at roots and we've looked at stems. And we've looked at the differences between monocots and dicots in terms of how they arrange their vascular tissue in their roots and their stems. So here's a question for you. So one of these is a stem picture and one is a root picture. Can you think of which one is the stem? So now you need to, excuse me, look at the vascular tissue and see how it's oriented in that cross section. And what you do is you look, you say, okay, is the vascular tissue closer toward the center of that cross section, or is it more toward the perimeter? And that will help you decide. So thinking about this, want to take a stab at which one is the stem, A or B? B. So think like an architect. If you're going to build a high-rise skyscraper, you don't want your steel girders in the center of that building. You want a scaffolding around the perimeter. And so B is stem tissue. So you have a stem coming up, out of the soil where wind is going to buffet it, storms, animals. You need to have some structural rigidity. Best way to do that is to put the vascular tissue, which has, that xylem. It's really structurally sound. So stick that around closer to the perimeter of your stem. So B is the stem. A is the root. You can see the vascular tissue is more in the center of what you see in figure A. So now we have to think about one of these represents a dicot and one of these represents a monocot. Which one is the monocot? So now we have to think about, again, the arrangement of the vascular tissue, the design of the vascular tissue. So monocot. Which one is a monocot? Figure A or figure B? Which one's the monocot? Is it figure A or figure B? A. It's A. So monocot roots have that ring of vascular tissue. Dicot roots, what kind of vascular tissue design do they have? It's not a ring. It's a cross or a plus. All right, so A is a monocot root picture. B is a dicot stem. Remember, it does that funky thing. When you look at the dicot root, it's that plus configuration. But then as it morphs into stem tissue, that plus configuration morphs into a ring-shaped design in the stem. So when you're trying to figure this out, you zoom in on the vascular tissue and you look, where is it in respect to everything else? Especially when you look at a cross section like this, right? So it's really trying to get down the design of the vascular tissue. There's differences depending on if it's a monocot or dicot or a stem versus. So we had that snow day last Thursday. So I guess our other snow day was a Thursday as well, I believe. So we have missed out on essentially a whole week of class lectures. So thinking about, we talked a little bit about the vascular cambium and how the vascular cambium let me go back and refresh your memory. So we talked about the vascular cambium. When we look at a dicot stem, you have that ring of vascular tissue. And in between the xylem and the phloem in that ring is the vascular cambium. And so that vascular cambium will go through cell division throughout the growth season, where you have that cell dividing to produce two progeny cells, and one becomes the xylem and one becomes the phloem. And over the years, you can get a buildup of xylem, secondary xylem, which are those annual rings that you can actually count how old that tree is by the growth rings. So that's secondary growth. It's secondary xylem growth. And you can see the phloem tissue, there's not as much of that because the older phloem tends to die off and is replaced by fresh new phloem. And so you really see this over time. You'll see that the tree has way more xylem than phloem. Yeah, and they can vary in size, like how much diameter to each ring, and that can give you a clue on what that plant was experiencing. Usually hardship, if they're under a tough year, the growth ring will be pretty tiny. And under a year of plenty where everything was going good, you'll have a nice big growth ring. And so, and then also growth rings, you can tell if there was some droughts going on or plenty of rain, because usually what you see is those really large vessels will get laid down early in the spring, and then you'll get the tracheids, which are much smaller in diameter. So this big swath here, those are all tracheids. And so it looks like in this growing season, they kind of had some drought going on there. If you are in a drought condition, you won't put down as much vessels. You'll go with the smaller diameter. It just makes it easier to get that scarce water up through your body without causing any air embolism in the tracheids during that process. So angiosperms will also factor that in. They will be paying attention to the water conditions and decide, so to speak, should I put down vessels or should I put down tracheids? So that's all the secondary xylem. So when someone's talking about the wood of the tree, it's really the secondary xylem that they're thinking about. And they often, if you're a forester, they tend to use the word springwood and summerwood. But again, the springwood would be the vessels of the tracheids, the vessels of the xylem. Those vessels with large diameter are really good at taking up really adequate water in the soil. They'll put vessels. Drought conditions, they'll go back to doing tracheids instead of vessels. And so those are the smaller cells. And then if you do take a cross section of a tree stem, you will see that there's discoloration going on, where you'll see that the inner rings are more dark typically. And so those are called the heartwood. That portion is called the heartwood. And then you tend to see the lighter portions. Often foresters will talk about sapwood. And so again, that's just, they're looking at older growth of xylem versus newer growth of xylem. And you'll see, I have some pictures here. And so the heartwood area of xylem, that secondary xylem that they call the heartwood, is typically pretty full of tannins and other resins. And so that is really not functional xylem. It's not really actively taking up water in that region. But it does help with structural support. And all of those tannins and resins also help protect the tree against pathogens trying to get in and start attacking. Those are good defense mechanisms. So here again, you're seeing some tree growth rings in the secondary xylem. And the vascular cambium is that ring of cells where you see this transition from the xylem into this area is the bark. We'll get into that. And so again, it depends on how well the spring is going. If you have a lot of rain, put down the vessels. If you don't have much rain going on, you get some drought, put down the tracheids. If you're an angiosperm, remember pine trees are gymnosperms. Gymnosperms don't really have that option because they only have tracheids to work with. And so gymnosperms will have just even look to their xylem cells. They're all tracheids. And so they look pretty similar. So the growth rings won't be so obviously different. And then here you can see a very dramatic heartwood versus sapwood. So the darker region is non-functional xylem in the sense that it's not taking up water and minerals, but it's still providing support, structural support, and defense. And then you see the sapwood. And then sometimes you'll hear people talk about hardwood versus softwood. Most of the time, gymnosperms fall into the category of softwood, and then dicot trees the hardwoods. So just in case you were ever wondering about some of those more common terms that get thrown around. And then the last thing I wanted to mention in this slide set that I wanted to explain a little bit more is the idea of bark. And so when you first see a dicot tree germinating and growing, a baby maple tree, let's say, or an oak tree, course, it doesn't have bark right away. It just has an epidermis surrounding on the surface of its body. So over time, there needs to be this transition from an epidermis to bark, and that is a extensive process. It provides the same basic function as the epidermis in the sense that it's the outermost layer, it's protective, and when you look up the definition of bark, that second bullet point is the definition. It's all tissues outside the vascular cambium is considered bark tissue, which includes the phloem. So phloem is part of the bark. And so remember that vascular cambium, it's that ring of cells that when it divides, it produces xylem to the inside, phloem to the outside. So everything from the vascular cambium out is bark, which includes the phloem. And so that's why you should never girdle a tree, where you take that bark and strip it off all the way around the whole tree diameter, all the way around the perimeter. If you do that, you remove all the phloem. Now the roots have no way to get sugars delivered to them. That's because the phloem's job is to deliver sugars and other important chemicals throughout the whole plant body. And so the plant will be busy doing photosynthesis, and then it starts sending sugars down to the lower parts of its body. And if you've girdled it, you've essentially created a very slow, agonizing death for that tree. They will die. It'll take them a while, but they will die. And so it can be very tempting, I know. When my boys were younger, go out in the woods and they didn't know any better. I'd see them on, the white birch trees with their bark tends to flake and peel, and it's so tempting to want to peel that all away. And I would have to distract them and tell them, no, you can't do that. You're going to kill the tree. And so you can remove bark from trees in a way that keeps them alive, but you got to know what you're doing. And so people get trained in. You can actually harvest. That's where we get cork from, like cork that goes into wine bottles. Not that you would know that if you're under 21, but the cork that you work with in wine bottling, for example, that's all coming from the cork oak tree. But they know how to harvest the bark to keep the tree alive, so it'll keep producing for the year. Yeah. So yeah, you can take like a section, and as long as when you girdle, of course, you're cutting off every, the phloem all the way around. Now there's no phloem, like the phloem will come, the sugars will come to that part, and then because you've girdled it, it just stops. And you can actually see it swelling right there because the sugars just land there, and then they're like, I got nowhere to go. You mean once it's cut off the tree and processed? It gets brittle, if that's what you're asking. But cork is amazingly water repellent. That's why they use it for wine bottling, because it makes it so that the wine doesn't get anything coming into it, any moisture to change the quality and taste. So cork cells and cork tissue, it's part of the bark. But there are plantations where they grow cork oak trees. I think I even have a picture in here. In a later picture I can show you. And so what they're doing is they're carefully removing portions of the bark. And when you look at the bark, it does include the phloem. We call it secondary phloem because it's being produced by the vascular cambium. So the secondary xylem, secondary phloem. So there's some older terminology in the botany world for different parts of the bark. So, paraderm is a continual term. We still use that term. Paraderm is what takes over and replaces the epidermis. So as that tree gets bigger and stronger and older, you'll see this shift from epidermis to paraderm. And then what happens is you'll see that paraderm start developing a layer called the cork cambium. So it's cambium. So they named it that way because that's another region of meristematic growth. So it's a ring of cells again, outside of the vascular cambium. It's in the bark areas. So we call it cork cambium, and it's going to go through cell division. And what happens is one of the cells will get produced to the inside of the cork cambium, and one will get produced to the outside. The outside cells are what we call cork. All right, cork cells. So as that cork cambium, that ring of cells is dividing, it's going to produce cork cells to the outside, and then we get what they call cortex cells to the inside. Those are just parenchyma cells. They're living, functioning parenchyma cells. So those cortex cells, those are called secondary cortex cells. They're just parenchyma cells. The older term for that is phelloderm, which I put in parentheses. You don't need to know that. It's just that some of the slides or images are labeled that way. And so I wanted to clarify. But I think it makes more sense if you learn that those cortex cells, it's just parenchyma cells that are getting developed. So bark is very diverse. As you know, if you've been out hiking around trees, there's all kinds of different designs out there and different amounts of thicknesses, and whether or not you'll even see the lenticels showing up in the bark tissue. This is not photoshopped, but this is a tree, a eucalyptus. It's called rainbow eucalyptus. And so this is a tree that's sadly found way south of us. We don't have the pleasure of seeing this when we're out hiking around in this area, but that's not Photoshop. Those are the real colors of the bark. Wouldn't that be awesome? I keep thinking we should try to grow this in the greenhouse and have it, because that is just stunning. And the other good thing about this tree, fun thing about this tree, is that it will peel off bark over time. So you get this kaleidoscope of colors through the life of that tree. It'll give you different color designs, and so it's really fun. If you've ever had the chance to see this in its native habitat, you're very lucky, because that's well south of here. But beautiful, beautiful diversity. And then you have, more like this, where everything is pretty thickened up and more plain Jane, so to speak, but still very functional and does its job. So here, what they're showing here are two images of early on is the top image, like a tree starting to form young bark. So it's early in its growth. And so the periderm is replacing the epidermis. All right, so you start to see this build up. And the cork cambium they have as this dark brown layer. So those are living cells. Okay, that's meristematic tissue that will divide and produce a cortex cell, which is a parenchyma cell to the inside, and then a cork cell to the outside. And then over the years, you tend to see this buildup again, similar to what we saw with the vascular cambium, get those growth rings of xylem. Now you see, several years of parenchyma cells building up, several years of cork cells building up. So you can get bark that thickens up like that because of the active cell division in the cork cambium layer, that ring of cells. And so it just depends on how old the tree is, how thick that bark will be. Here you can see, if you look at the bottom of the image and you start working your eye up, you can see how they kind of look brick-like. Those are all the cork cells. Okay, so it's very thick layers of cork cells. This is a pretty older stem tissue. And then you get into the cork cambium area. You can see where you start to see these plumper cells. Those cells are the cortex or parenchyma cells, right? They're plump, they're living, doing their thing. Here's another image where you can see again, the cork cambium. Some older texts will call it phellogen. I don't really want you to learn that term. Learn it as cork cambium. And then the cork cells, and then the parenchyma cells. Okay, so it's just this other next layer. So this is a nice cutout where they're showing both ideas here, where you have the vascular cambium is right where you can see my red laser pointer. So that's the vascular cambium that's dividing to produce secondary xylem to the inside, phloem to the outside. And then if you work your way out into the bark, the cork cambium is further out. Okay. And so again, that's another meristematic region that allows for parenchyma cells to the inside, cork cells to the outside. Okay, so you have actually, when you look at a more mature tree, they'll have those two distinct rings of vascular tissue, the vascular cambium and the cork cambium. And they are necessary to get that nice, healthy stem tissue and then the bark tissue covering all of that. And then don't forget, we learned about lenticels in lab. So bark will have lenticels. You can't always see it though. Some bark tissue, you just can't see the lenticels with the naked eye. You have to take some samples and look under the microscope. But again, the cork cambium is producing that. It's part of the bark. And so you have, again, all the living cells in the stem have to get access to oxygen to carry out respiration, just like you need oxygen to carry out respiration. So the lenticels will provide looser patches of cells to allow for a little easier flow of oxygen to get in. You'll find it on apples and pears as well. So those are really easy. If you look closely before you bite into them, you'll see the lenticels. So lenticels, again, sometimes they're visible and very obvious, and other times not so much. And then under the microscope, you see that loosely packed region, discrete region. Any questions about bark tissue? Yes. Yes. You got it. Yep. So it's a gradual transition. First you have the baby seed, then the seedling, and then eventually you'll start to see the bark starting to show up. It takes a while. Depends on the plant, I suppose, but most trees take a while for that to happen. Okay. The last set of slides in this slide set are just looking at modifications to stems that plants will do. And so, looking at tendrils, for example, that's actually modified stem tissue to be more flexible and have a way to wrap around some kind of support so that they can keep themselves upright. You have some stem tissue that's very squat and short, and it creates what we call rosette plants because the stem is so short, all the leaves are closely attached and they give this rosette pattern when you look down on it. Some have modified stems that have shifted into thorn tissue. So you have stem tissue that can modify itself. Remember, underground stems, we call those rhizomes. So ginger rhizome is being shown here. The white potato is actually stem tissue. So when you eat a white potato, you're actually eating stem tissue, modified stem tissue. And the skin of the potato is actually very thin bark. You're eating bark tissue. It's just not super thick. And you should eat it, by the way. Wash it and eat it because it's very healthy. There's a lot of nutrition there. Don't eat this though. This is what I study. One of the diseases of plants that I study is called potato scab disease. This is a very severe example of it. You would not see this in a store anywhere. You could eat this if you wanted to. I have on a dare. I did eat it. I don't advise it because it tastes like wet dirt. It tastes like you just put a bunch of dirt in your mouth and are chewing it. It tastes like wet dirt. Now I love the smell of wet dirt, but I don't love the taste of wet dirt. So it's not dangerous. This pathogen isn't dangerous to you, but it's certainly, as you can see, very dangerous to the white tuber, the potato plant. Corms and bulbs. Remember that there is a definite difference. When you think about corms, it's a lot of stem tissue, very little leaf tissue. The leaves are thin and papery, and then bulbs are just the opposite. Very little stem tissue, lots of fleshy leaves. And so again, some modifications. And then don't forget, there are really important things that we use from stem tissue for human uses and other uses. So stem tissue, of course, for building and lumber, but also there's a lot of other reasons. So think through some of that idea. And then this is the cork oak I was telling you about, the cork oak tree, where you can very carefully harvest portions of the bark. So you see here, they have a cork oak plantation, and they are, that tree is obviously alive. It's alive and kicking, and yet they're harvesting. So I have not seen this done in person, but it's doable. Amber is coming from the secretions of stem tissue, some of the secondary metabolites, which of course can get fossilized over time and have been the subject of a lot of movies, and very beautiful things can get captured. Here you see an insect captured in the amber. And then the bristlecone pine, it has a very torturous looking stem tissue, but it's argued to be the oldest living organism, although there's debate over that. But this one is in California, called Methuselah, and they think it's over 4,700 years old. That's old. That's impressive. They don't have this on any kind of tour. They don't want people finding this one because people in the past have taken samples from it and have risked killing it. So it's now top secret of wherever it is. I have not seen it myself. And then of course, massive trees. You think about their stem tissue. That's not me, unfortunately, but I did get to go see the giant sequoias in Yosemite. There we go, Yosemite Park. And I would highly recommend that. I did not get a chance to go and see the giant redwoods, but that's the next one on my list. So these are giant redwoods, and just stunning. You can see the size comparison of the people here. I did not take these. A student in this class gave me these pictures when she went. So stem tissue, lots going on there. Lots of beauty as well as very useful things going on. Okay, so that was the last of the stem tissue section. I wanted to make sure to go through the bark information in particular. And so the next thing I'm going to get into right now will not be on this exam because it will be going into the leaves. Did I put those up in EduCat yet? Okay, the leaves. So let's shift over. So there's two slide sets on leaves, primarily because there's a lot to say about leaves. Leaves are incredibly diverse. When you look at the plant body, leaves are the most diverse in terms of the design. The morphology of leaves vary the most compared to if you look at roots or stems. Leaves have way more diversity in how they look. And so when you think about the leaves can be just a couple millimeters big, where you have to squint to look for it and find that leaf, up to, you know, really large leaves, aquatic leaves like this. The giant water lily, you might have seen video where they show animals walking on it. There's even one of a little kid sitting on it and it stayed upright. So there's a lot of structural, architectural support under these leaves. Even though they're quite big, they can maintain that upright, floating posture. And then there's really big terrestrial leaves. You can see here with the size difference, it's the person's head in the middle of the leaf. And so this, the Gunera species, giant rhubarb, sometimes it's called, a dinosaur food. I like that one. Here's what it looks like in a landscape design. I don't think it would do so well here, although I know of one place in town where it looks like they have a patch of this in their front yard. And I always drive by off and on through the summer to see how it progresses. And it's not this big. It doesn't get this big. I think it's too cool up here, but it can get impressive. So leaves, you learned a little bit about leaf design in terms of how the leaves attach essentially to the stem or branch tissue. And so alternating just means it's every, on either side and kind of does that Z pattern on up. If they're directly across from each other when they attach, that's called opposite. And then if you look at the design and it follows more of a spiraling up attachment pattern, especially as you look down on it, they'll just usually say spiral or helical. Whorled is something a little different where you have 3 leaves. Okay, it's reserved for this idea that you have 3 leaves attached at the same point. So we say it's whorled. I don't really get into decussate, but that is another one. And so this is something, I know it's like, more terminology, but it's something that we look at when we're trying to identify. So if you buy a field guide to trees, for example, this is something that they'll ask you to look for. They'll say, what's the leaf attachment? And then that will help you figure out what species of tree you're looking at. And then you have this idea of, are they simple or compound in the leaf design? And so the way to, so compound palmate, what they mean there is palmate is actually referring kind of like to the palm of your hand. So if you think about the point of attachment as like the center of your palm, and then all the leaves coming off of it, kind of like your finger. And so the palmate design. And then you have compound pinnate, where they're more alternate or opposite attachment here. And so you get those types of designs. And let me shoot to the next slide, because when you are trying to figure out, well, I don't know, is this a simple or is this a compound leaf? What you should look for, if you can see it visually with the naked eye, you look for the axillary bud. If you can find that, then you can figure out if it's simple or compound. So the axillary bud here. So if you see this leaf, and then the leaf petiole coming down and attaching, and then you see an axillary bud right there, this would be a simple leaf. Now, if you compare that to the right image, if you look at what this, you're like, oh, that looks like a leaf. Here, let me use my laser. So if you look at this one and you go, oh, look at that leaf, but you don't see an axillary bud anywhere here at the base of this leaf. So technically this isn't a leaf. It's a leaflet. So that's what they have here, leaflet. So the trick is find the axillary bud. And so on this one on the right, the axillary bud's way down here. It's not at the base of each of these leaflets. If it was, then we'd say that's a leaf. But because the axillary bud is all the way down here, what that is saying is this is the leaf petiole, and all of these are leaflets. So taken together, this whole right image, the whole thing is a compound leaf. Whereas over here, this is a simple leaf. This is the leaf, because if you follow that down where it attaches, you see the axillary bud. So that's the trick. So again, this is well labeled here, looking at where's the axillary bud. And so again, field guides, if you're trying to identify a tree, they'll ask you to look for this. And then they'll use the terminology a leaf versus leaflets, simple versus compound. So here you can see that each one of these is a leaf because the axillary bud is right there. So each one of these is a leaf. Okay. So again, the axillary bud, remember, that's meristematic tissue. They're just not, they're dormant. That tissue is like quiet right now. Because remember, auxin is at the shoot apex, and auxin is coursing down the stem, telling those axillary buds to be quiet. This is important to realize too. So if you are out hiking around and you see a young seedling, that can be really hard to identify what it is, because what you're seeing here is the exact same species of plant. But on the left, that's what it looks like when it's a baby, when it's very young. And then as it grows older, its leaf design morphs into a very different design. And so this is the same species. It's just, we tend to say the young ones are juvenile, and then the mature plant we say is the adult plant. And so that can be very confusing. So I have a hard time with very young seedlings when I'm out hiking around trying to figure out what they are, because it can be tricky. And then, of course, the shape, the overall shape, and they look at the margin of the leaf. They look at whether or not it's lobed. They look at the vein pattern. So all of this is important if you're trying to identify a plant. You learned a little bit about vein pattern in lab, I think, where and we mentioned this last week, I guess last Tuesday, we're talking a little bit about this, where you look at the veins on the leaf, and that can help you decide if you're looking at a monocot versus a dicot. So most dicots have the netted veination, where it's very, the veins are going every which way. If you look at it carefully, it does look like kind of like a fishing net, whereas monocots tend to have that more parallel veination pattern. It's not 100%, but it's a really good rule of thumb to follow. And so look for that as well as you're trying to identify, get to know your plants. So leaf veins, when you think about the leaf veins, we're looking at the vascular tissue, of course. So let's zoom in here. We talked a little bit about this last Tuesday, I think, where we were saying how inside the leaf, you can have differences in design too, where you might have multiple palisade layers, maybe just one, maybe two, maybe three. You might have spongy mesophils. Sometimes it'll look very much like this, where you see very different palisade and spongy parenchyma cells. Other times it looks more like, oh, it just looks like it's all spongy. So there's design differences inside the leaf too that you need to think about. Of course, the veins, the vascular tissue, and then depending on where the stomata are placed, that can tell you a lot about how that leaf spends its time with orientation to the ground. So we'll talk a little bit about that again. So again, you've looked at this in the lab under the microscope. You can see epidermis layers can be very puzzle or jigsaw-like. Sometimes they're brick-like, they look like a brick wall, but all of them will have some kind of stomata scattered across to help with intake of carbon dioxide and oxygen, outflow of water vapor. So remember, those guard cells are really well designed and very tightly controlled by the plant. The plant has really good control on whether or not those guard cells reveal the pore or close. And again, it's all being driven by what they're sensing in terms of their water status and the water availability in the soil. And then don't forget, as soon as the stomata open, you have transpiration happening. So it can't not happen. It's just automatic. If the stomata open up, you're going to have water leaving the plant body. It just happens. Do you remember what force is actually causing that to happen? Is it pressure or tension? Positive pressure or tension? Turgor pressure to get the guard cells to open, for sure. So turgor pressure is actually allowing those guard cells to move away from each other and reveal the pore. You get a flow of water into the guard cells, and that causes the pore to be revealed. So as soon as that happens, water is going to start exiting the leaf as water vapor. But do you know the force? As water is leaving the plant body, what force is driving that? To the positive pressure? Is water being pushed up the plant, or is it being sucked out of the plant? Sucked out. So that's tension. Tension is negative pressure. Tension is sucking action. Think about, sipping soda through a straw. All right, so that idea, and so it just automatically happens by the idea of the cohesion tension theory. And so it's a risk. Anytime a plant opens up its stomata to get access to CO2, it's risking water loss. It's going to happen. And then they have to constantly assess, okay, am I still okay with my water status? Am I getting dried out? And then they have to make a decision. Even if it's during the day, they might have to shut down their stomata if they're drying out too much. And then, of course, they can't take in CO2. So it's this compromise is what they're referring to. They have to compromise. Like, I can't lose too much water, but yet I need CO2 to come in. And so it's this constant assessment of those two ideas. So we've already talked about transpiration last week, and I put it in this slide set too, just to make sure that you're still thinking about this idea because again, leaves, it's super important process going on in the leaves is that water vapor that is in the leaf tissue will start exiting out of the leaf as soon as the stomata are open. So you've seen this already. I've showed you this in the root slide set. I just put it here to make sure to remind you to think about it for when we get into the leaf processes as well. So we looked a little bit at this idea last week, how leaves and cross section, again, when you're looking at these, looking at the different types of leaf cells. So you have the palisade cells up top here, and then you have the spongy cells, spongy mesophyll cells on the bottom. They're more round, and then all those air spaces for water to evaporate into. And I told you last week to imagine cutting that in half inside the leaf. So if you take and just kind of cut the leaf in half, can you see my red line all the way up there? Okay. And then you look what's happening above the red line in terms of what kind of cells and how does it look above the red line, and then how does it look below the red line. And in this case, I think you could come to the conclusion pretty quickly, they look different, right? What you see above the red line is those long, slender palisade cells. What you see below are those fatter, spongy mesophyll. So it's not symmetrical. They look different on either side of that red line. That's a giveaway that that leaf spends more of its time horizontal to the ground. You can also look for the stomata as well to further lend you some clues. And you can see this isn't the best prep, but there are some stomata on the bottom. There's no stomata on the top. And that makes sense too. If you spend your life more like this, tuck your stomata in the shady part of your leaf. You don't want all your stomata to get hit with bright sun that makes you lose water quicker out of your body. So tuck them under, get it where it's shadier, cooler, more humid. That will slow down water loss out of your body. We also found out that some trees can do both shade leaves and sun leaves. So the top one is a sun leaf. So a maple tree, its very top leaves will look more like the top picture, where they're thicker, they're smaller in surface area, but they're jam-packed with usually multiple layers of palisade cells. And then this bottom image is off the same tree. It's just the shadier portion of the tree. And again, they tend to be a little thinner and a little bit wider in surface area, trying to get access to that very sparse sun. And then if you see something like this, again, we looked at this already, but I think it's good to reiterate. So again, cut this leaf in half and look at what's on the top versus what's on the bottom. And they kind of look like mirror images, right? I mean, if you look what you see here, and then you look below, it's pretty much the same. You're getting pretty much the same design. And so that's a dead giveaway that that leaf spends more time vertical to the ground. And then you look for stomata and you see that there's stomata on both this top portion of the image and the bottom portion of the image. So again, if you spend your life like this, you probably have about the same amount of stomata on each side of your leaf, because you're going to get hit, this side gets hit with, that's north, south, west, east. So I guess the sun rises this way for you if you're looking that way. So it's going to get hit with half sun on this side, half of the day on this side. So it doesn't matter where you put your stomata. So it's kind of interesting when you look at leaves under the microscope, you can actually get quite a bit of information about the life of that plant by looking at its design inside. And this one's a good one because if you look here at the bundle sheath cells, you see how much more plump they are. So the bundle sheath cells that are surrounding the vein, the xylem and phloem, they're plumped up. And that's a dead giveaway that you're looking at a C4 plant because they have plump C4 cells. They got so much going on in there that they really plump up. You'll see floor plans. So we've looked at those in lab, so I won't ask you any specific questions on the lecture portion with regard to leaf tissue, but remember, you'll probably get a 2 or 3 questions in lab from what you did in lab, okay? So remember now, Thursday is exam day in your lab, but I will hold lecture session as well. So we will continue talking about leaves on Thursday, okay?